Hi, I'm Spencer Krieger, and I'm excited to share work I did with my PhD advisor, John Kececioglu, titled Computing Optimal Factories in Metabolic Networks with Negative Regulation. Factories are a representation of pathways which are a collection of biochemical or physical reactions that culminate in a specific response. A simple example is the insulin pathway, which results in the storage of blood glucose in the cell. First, insulin docks over here on the right in the insulin receptor, which causes a cascade of internal reactions, here notated by 2, opening the glucose transporter, notated by 3. This allows glucose from the bloodstream into the cell, where it can be stored as glycogen or converted into fatty acids, as shown by the reactions labeled 4, 5, and 6. A metabolic network is a collection of reactions from many different annotated pathways, all concatenated together. Here I have a picture of a metabolic network that is the combination of the gene regulation, cell proliferation, and apoptosis pathways, with receptors around the outside of the cell acting as sources. For many reactions in a metabolic network, the stoichiometries of each participant in each reaction is also recorded. A collection of the metabolic networks of many different organisms is called a network database. We present solutions in this presentation to the pathway inference task, which is, given a set of available source compounds, think of these as things present around the cell, and a set of target molecules you would like to make, we want to find the series of reactions that produces the targets from only the sources. So why do we care about inferring pathways in metabolic networks? Pathway inference can identify crosstalk, which is explained in the following picture. Pathway 1 is an annotated pathway from S to T, and pathway 2 is an annotated pathway from S' to T'. By inferring a pathway from S' to T, we can show that the activation of pathway 2 can cause partial activation of pathway 1. A second purpose of pathway inference is identifying therapeutic targets. Given a disease symptom T, we can infer the pathway from our sources, S, which identifies a protein represented by vertex V as a potential therapeutic target for the treatment of this disease. So pathway inference can be important in both identifying crosstalk and identifying potential therapeutic targets for disease. Now that we understand pathway inference and why it is important, I next want to describe how we model metabolic networks for pathway inference, starting with the reactions that make up the network. Here's the general form of such a reaction. On the left, shown as A and B, we have the reactants, which I've colored in blue. And on the right, we have the products, which I've colored in pink. The small letters represent the stoichiometry of the reaction, which are positive integers that tell you how much of each substance is used or created in the reaction. Here I've shown them in green. Positive regulators promote a reaction, where negative regulators inhibit a reaction. Importantly, reactions require that all the reactants are present, and then all of the products are produced when the reaction takes place. To infer pathways in these networks of reactions, we want to represent these reactions as edges in graphs or hypergraphs. Traditionally, given this reaction from the previous slide, each reactant or product would be modeled as a vertex, and then each reaction would be modeled as a collection of edges from each reactant to each product. Notice how traversing any of these edges does not require that all the reactants are present, since the edge from A to C can be traversed without any constraint on B. This reaction as a single directed hyperedge, where all reactants are in the tail of the hyperedge and all products are in the head on the right. Notice how now hyperedge E can only be traversed when all of the reactants are present and all of the products are reached or produced upon the traversal of the hyperedge. I now want to introduce the idea of metabolic factories, which are a hypergraph model for pathways, which specify a flux for each reaction in the network. Flux is the relative usage of a reaction in a metabolic network and is represented by a non-negative real value for each reaction. All intermediate metabolites, which are not sources or targets, must either satisfy conservation, which here you want the influx to equal the outflux, which is the total flux on incoming edges equaling the total flux on outgoing edges, or you like intermediate metabolites to satisfy accumulation, where the flux in is at least the flux out, which captures that the intermediate metabolite is not depleted. Factory specifies a flux on each reaction in the network that produces all targets from sources and satisfies either of conservation or accumulation for all intermediate metabolites. Last notion that I want to introduce is negative regulation. So this picture here shows one of the ways that negative regulators can inhibit reactions. The top row shows the reaction proceeding normally, with the substrate binding in the active site and then the products being released. The bottom row shows the inhibitor blocking the active site, stopping the reaction from occurring. Prior pathway models have not considered negative regulation, which we consider for the first time. To preview our contributions, in our paper we introduced the new problem of finding a factory from sources to targets using the fewest possible reactions, and we proved that the problem is NP-complete or computationally difficult. We handle negative regulation for the first time in pathway inference, considering direct and indirect effects. Our approach based on mixed integer linear programming finds optimal factories remarkably fast, with a median runtime of only 3 seconds, even including the first and second order effects of negative regulation. Our approach is available in the tool Odin at odinn.cs.arizona.edu. I will now show the two orders of negative regulation that we have modeled. First order negative regulation is direct negative regulation. We call a given factory first order negatively regulated if a vertex in the factory, here R, negatively regulates another edge in the factory, which invalidates the factory. Second order negative regulation is more indirect. Here, if a vertex R is reachable from the active sources, and that vertex negatively regulates an edge inside the factory, we consider this factory invalid because we know that the sources are active, so R could be created and inhibit the factory. To versions of the min-edge factory problem under 0th, first, and second order negative regulation. I will now move on to experimental results. We constructed hypergraphs using human-curated metabolic networks from standard databases, Reactome, and organismal networks from MetExplore. I will now give a concrete example of how we would map the formation of this complex A/B/L to a hypergraph. We represent each protein and small molecule, like ligand L, as a vertex along with the A/B/L complex. We then map each reaction to a hyperedge. So this reaction maps to this hyperedge. Just a brief summary of our results on negative regulation. We found that no optimal 0th order factory, or one that does not consider negative regulation, satisfied first order negative regulation for 83 of the 4,000 reachable target instances. It demonstrates the importance of considering negative regulation, as otherwise you find non-viable factories. To give a more concrete example of 0th and first order negative regulation, we have this example. Here, the 0th order factory has 17 hyperedges, but uses a hyperedge that is negatively regulated by one of its products, GSH. Instead, the first order factory, shown in green, uses 21 hyperedges because it must avoid using the negatively regulated reaction. Our implementation of our mixed integer linear programming-based approach, Odin, is remarkably fast in practice, with a median running time, including both orders of negative regulation, of only 3 seconds. The maximum running time is also fast for 0th and first order negative regulation, being just over a minute. To conclude, we introduced the new problem of finding a minimum hyperedge factory. We incorporated negative regulation for the first time into pathway inference. We showed that we can compute optimal factories remarkably fast in practice. And an implementation along with all of our data sets is available at odinn.cs.arizona.edu. I would like to thank T.M. Morale, Anna Ritz, and funding from the National Science Foundation. Thank you for listening to my presentation.